This video is going to cover Unit 1 and the very first part of Unit 1 of Higher Human Biology, so the Division and Differentiation of Human Cells subtopic. So this is the very first one that you're going to cover in the whole of the course. You should know the cell structures and the cell functions, but this is the main initial part of the course. So it starts off with the two types of body cells that we'll need to know, and the very two general types of body cells we'll need to know. So you need to know that there are somatic body cells. So these are body cells that are not involved in reproduction. Okay. Now these somatic body cells are what is known as diploid cells, which is a term that we should know from National 5. Okay. So when it comes to higher, you need to know slightly more information than that. You need to know that diploid cells have 23 what's called homologous chromosomes. Now homologous just means matching. Okay. So chromosomes that are matched as pairs. Okay, now because there are 23 pairs of chromosomes, 23 matching pairs of chromosomes, that means that we've got two sets and ultimately 46 chromosomes. Okay, now somatic body cells will carry out the type of division that we know from National 5. So they carry out mitosis. And that mitosis, just like in National 5, will then produce more diploid cells. Okay, and specifically more diploid somatic cells, more diploid body cells. Okay, so these types of cells are things like liver cells, skin cells, neurons, any type of body cell that is not involved in reproduction, so not the gametes and not the cells that produce the gametes. Okay, and just to point out a wee diagram here, these are chromosomes down the bottom. So this is a chromosome and this is its homologous pair. Okay, so it's homologous matching chromosome. Same with this one, although it's smaller, this is a chromosome and this is its matching pair. So when we talk about homologous chromosomes, we're talking about ones that look similar. They'll have genes in the same place. Okay, they'll have roughly the same size, they'll look roughly the same shape, and they're just generally matching. Okay. So that's somatic cells and they would divide by mitosis. So for germline cells, these are the cells that are produced, that are used in then reproduction, so the gametes, and then the cells that also produce the gametes. So those cells that are in the testes and the ovaries that would produce the sperm and the egg. Okay, so that's what I've got down here in this wee diagram. Now this sperm and this egg are, specifically we're actually going to call it an ovum now, and higher we call an egg an ovum. Will each then have their own chromosome complement, which is different to this one we talked about in terms of diploid cells for the body cells. Okay, so initially germline cells are diploid. Okay, so the cells that would produce the gametes in those testes and ovaries are diploid. But they can divide to produce either copies of themselves, which are diploid, or they can produce the gametes. Okay, so they can divide to produce haploid gametes, through what we call meiosis, which is a new type of division that we don't know about yet. And they'll also be able to produce diploid germline cells, so more copies of themselves, with the same processes we talked about here, so through mitosis. Okay, so when we're talking about haploid gametes, then these have one set of chromosomes, okay, each, and therefore 23 chromosomes, so that when they combine and the nuclei of each of them combine through fertilization, then we get a full diploid zygote. Okay, so to give you a bit of overview of the divisions and a bit of a reminder from National 5, I'll talk about mitosis first. So mitosis only involves one division. So if you think about mitosis from National 5, we'll have chromosomes replicating, your nuclear membrane will disappear or it'll break down, and then the chromosomes will line up along the equator. Now what will happen then is the cell divides ultimately, right, once those chromosomes are separated, or the chromatids are separated into chromosomes, and then you produce a new cell that is diploid here, okay, and a new cell that is diploid here, okay, and this original cell is diploid because only diploid cells will be able to divide by mitosis. Okay, and if you count the number of chromosomes, we've got 1, 2, 3, 4, even though they're paired together and they form those wee X's, right? This is still counted as one whole chromosome. Okay, and this is one whole chromosome. So we have 4 chromosomes here, and then this cell has 4 chromosomes here, and so does this cell. So we've not half the number of chromosomes we've got, they're the same number. Okay, so mitosis is carried out by both somatic cells to produce more somatic body cells that are diploid, and they're also carried out by germline cells to make copies of themselves, those cells that are in this, the testes and in the ovaries. Okay, so that's mitosis. You start with a diploid cell and you end with 2 identical diploid cells, each with the same number of chromosomes. Now meiosis is different to this, and you won't need to know it in a huge amount of detail, but there are 2 divisions in this that you will need to know. Okay, so it involves 2 divisions, and it's only carried out by germline cells. It is not carried out by somatic body cells. Okay, because body cells want to maintain their diploid chromosome complement, they want to stay diploid. So it's only carried out by germline cells. Okay, and it involves 2 divisions this time. So just the same as mitosis, okay, we start with our chromosome replication. So these chromosomes have duplicated and formed their wee X's. Okay, and then in this stage what happens is we have a diploid cell, okay, with 4 chromosomes. That's its chromosome complement. It's diploid and it's got 4 chromosomes. Now what happens here is these matching chromosomes are separated. Okay, so if we colour this one in red, and the same with this wee guy, these 2 are pulled to here. Okay, and the other 2 are pulled to here. Right, so what's happened in this stage is then that the homologous chromosomes, those matching chromosomes, have been separated. Okay, so the ones that are the same size and the same shape and things like that, they pair up together and then they are separated first. So you get a large one and a small one here, a large one and a small one here. Now to form your haploid gametes, what has to happen is these chromatids have to separate, and so do these. Okay, so in this stage then the chromatids separate. Okay, so if we look at this cell to begin with, it is diploid and it has 1, 2, 3, 4 chromosomes. This cell now has 1, 2. Okay, and this cell has 1, 2. And then what happens is once we separate the chromatids, you get these haploid gametes with only 2 chromosomes. Okay, because they're half the number of chromosomes that that original diploid cell was. So these now are haploid. And they're gametes because they've got a little tail, they're a sperm. Now it could be an ovum here, but just for the purposes of a diagram, I've just made them a sperm to make it easier to see. Okay, so there are 2 stages involved in meiosis. Homologous chromosomes separate, so these matching chromosomes are separated into 2 cells. And then after that, the chromatids separate. So we pull these wee chromatids apart, and these then become chromosomes again. Now I understand that the phrasing of this is really difficult. The only time you ever need to use chromatids is when they're separating these right down the middle, okay, and you get 2 wee separate wee lines. Okay, they're always called chromosomes, right, when they're separated. Okay, so these gametes then are haploid cells that are used in fertilization, right? So again, this is a National 5 point that you would know. So they're haploid cells and they're used in fertilization. So the nucleus of an ovum, right, which is now what we would refer to previously as an egg, but we now refer to as an ovum, will fuse with the nucleus of a sperm. Okay, so here is our wee sperm here, which is a haploid cell. Okay, when humans we have 23 chromosomes. This ovum is a haploid cell with 23 chromosomes. And then what happens is the nuclei of these two fuse together, and then we get what is called a diploid zygote. Now this process here that has happened is fertilization. And fertilization results then in a diploid zygote. Now for this diploid cell, this diploid zygote, to form this bundle of cells, which is called an embryo, okay, which is also diploid, what has to happen is those cells have to divide. So it would divide in half, okay, or well, it would duplicate really, and form 2 cells, and then 4 cells, and then 8, and then 16, 32, etc., etc. Okay, so that process is called mitosis, where a diploid cell produces more diploid cells, okay, and that would form that embryo ultimately. Now this embryo then, to start to form particular areas of the body, because it's going to ultimately become a person, would then start to have to specialise those cells. So an area here would maybe form a leg, an area here would form an arm, an area up the top would form a brain, or whatever. Okay, so we have to then undergo what's called differentiation. Okay, which is ultimately what we used to call specialisation. Okay, when cells become more specialised. Okay, so to talk about differentiation then, in National 5 we refer to this as specialisation, so cells becoming specialised. Alright, so differentiation is just a fancy term for this, is cells becoming specialised. Now that happens, cells start to develop their own particular functions because they express genes. Okay, so remember that genes are short sections of DNA on a chromosome. Those genes are expressed and then they produce a certain type of chemical. If you think back to National 5, the DNA within genes, right, and those genes themselves are then containing instructions to make a particular chemical. And that particular chemical is proteins. Okay, so if you would switch on or off a gene, it would then produce or not produce a particular protein. Okay, so the example I've got here is a cell in the bone marrow. Now your bone marrow is the site of production of all of your body's blood cells. So red blood cells, particular white blood cells, platelets and other things like that, okay? This cell in the bone marrow, which is a stem cell, which I'll come on to, will express the genes to produce a particular protein, right? And in this case, we're going to say it is hemoglobin. Okay, so that hemoglobin then is produced, and this cell ultimately differentiates to form a red blood cell. Okay, that is differentiation. It is expressed genes that then produce a particular protein that then allow this cell to become specialised. Okay, another example here is that a cell in the pancreas would express a gene, and that then would produce a particular protein called insulin. Okay, and therefore this cell would ultimately begin to produce insulin, right, because it is producing that protein, and it would form a beta cell, what's called a beta cell, that would produce then that protein insulin. Okay, so differentiation is basically just your cells expressing particular genes to then cause the production of proteins that would make them specialised to suit a particular function. Okay, now it said it was going to come on to stem cells, which is what I've got here. Now stem cells, what you know from National 5 is that they are unspecialised cells. They do not have a particular function. Okay, those stem cells will then divide either to what's called self-renew, right? And if they self-renew, it essentially means that they make copies of themselves. Okay, or what they can do is they can divide to then allow them to differentiate, okay, so to become specialised into those specialised cells. Okay, so they can either self-renew or they can divide to then produce cells that would be able to differentiate. Okay, and as a reminder, stem cells are just general body cells. They're not involved in reproduction mostly. Okay, so they're mostly diploid cells. Right, so there are two different types of stem cell mainly. There are technically a little bit more than that, but we'll only focus on the two main types that you should know for hire. Now, one of those is embryonic stem cells, and they're found in the embryo. That bundle of cells we talked about at the end of fertilisation that's produced after a zygote will divide by mitosis and form a ball of cells. Okay, now these embryonic stem cells are what is referred to as pluripotent. Now, pluripotent basically means that they can become and they can specialise and differentiate into any type of cell. Okay, any type of human cell in the body. Right, they have what's called an unlimited differentiation potential. Okay, that means that they can become any type of cell in the body. So if you used embryonic stem cells for research, for example, they could then be allowed to differentiate to become any type of cell that they could then test drugs on, for example, or other things that we'll come on to in a minute. So all the genes in these cells have the potential to be expressed. Okay, so that basically means that they've not all been switched off or they've not all been switched on, for example, and therefore they've not begun to express particular proteins yet. We can flip a wee gene on and then tell it to produce a certain type of chemical. Okay, so these have an unlimited differentiation potential. They can become any type of cell. Okay, and that's what is known as pluripotent. Okay, now tissue stem cells are the ones that are found in adults. Okay, and there's quite a number of locations in your body that tissue stem cells are found. They're found in the skin, the intestines, in your heart, for example. But the main one that you will need to know that I'd referred to earlier on was in bone marrow. Okay, now the cells in bone marrow can then differentiate to make your red blood cells, your white blood cells, platelets, which is the things that help you clot, and any type of blood cell within your body. Okay, so the bone marrow is the stem cells that produce all your types of blood cells. It can't produce skin cells or muscle cells or neurons. It'll only produce the cells that are specific to that tissue type, to that bone marrow type. Okay, now these tissue cells are involved in growth, repair, and renewal, which I've actually just written underneath, so I don't know why I'm rewriting it. Okay, and these are what is called multipotent. Now, multipotent is different from pluripotent. Multi essentially means many, but it doesn't mean every type. Okay, so these can only become the cells of their tissue. Okay, so therefore they have a limited differentiation potential. Okay, so what that means is they can only become a very limited number of types of cells. So if it's within the bone marrow that these stem cells are found, they can only produce blood cells. If they're found within the basal layer of the skin, then they'll only produce the type of cells that are found within the skin. Okay, so they're very limited in terms of the number of different types of cells that they can produce. So this is because some genes have already been switched off. Okay, so they've already started that differentiation pathway. Okay, they're limited to the types of cells that they can now produce because you've kind of limited which pathway they can go down. Okay, so stem cells are fascinating because they can be used in research, right? In therapy and in research. So the therapeutic uses of stem cells are ones that we can use for treatment right now. Okay, so right now we can actually use stem cells to grow replacement corneas. Okay, so rather than having a corneal transplant, we can grow replacement corneal tissue for people. So this is a cornea, so this is your eye, and this is the lens. Okay, and this is the back of your eye where your optic nerve is. This is the cornea, which is the bit that protects the front of your eye. So if you've ever scratched your cornea, you'll know about it. Okay, it's very painful, but it protects your eye, prevents anything getting into the pupil and whatever else. So it's a layer protecting the front of your eye. Okay, if it gets damaged, it can then essentially cause people to become blind. But if it's replaced, then you can allow people to see again. What can also happen is we can regenerate damaged skin. Okay, and that allows for us to provide skin grafts then for people with burns. Okay, there's other potential uses or uses that have been trialled so far. So allowing repair of spinal cords for people with spinal cord damage. But the main therapeutic uses at the moment are replacement corneal tissue and then regeneration of damaged skin. The other use of stem cells is research-based. So it's what we could potentially use in the future for therapies but haven't been proven to be useful yet, or for other research testing. Okay, so these cells can be grown and cultured in a lab to produce model cells. Okay, and those model cells can then be used for a couple of things. They can be used for drug testing, so that we don't have to test them on live humans until we know the effects of them. Or they can be used for research purposes, and particularly for research purposes into disease development. Okay, so we can study how diseases develop to better understand it, to then allow better treatments of it. Okay, but the one issue with this, or the main issue with this, is the fact that it involves the destruction of embryos because we use embryonic stem cells. And that's controversial because some groups see that as destruction of a life. So it's something to be taken into consideration when we use stem cells. Okay, and finally, we're on to the very last part of division and differentiation, which is to do with cancer cells. Now, this is because we're talking about division of cells and cells within the body, and ultimately sometimes division of cells in the body can go wrong. So if cells in the body don't respond to what's called regulatory signals that tell it to stop dividing, okay, so cells are told to stop dividing sometimes. If they are then not told to, if they don't respond to those instructions and they don't respond to those regulatory signals, they divide what is known as excessively. Okay, so you end up with a huge number of cells that you shouldn't have had. And that forms a massive abnormal, right? So not typical cells, which are cancerous cells. And that then, when you get a lump and a mass of those abnormal cells, it's called a tumour. Okay? Now, that tumour, then the cells within that tumour tend to lose the wee bits on the surface of their cells, the molecules on the surface of their cells, that then allow them to attach to the cells next to them and hold on. Okay? But if they lose those molecules, they fail to attach to one another. Okay, so they fail to attach to the cells around about them. And when they fail to attach, then they might break off. And if they break off, then they spread throughout the body. And if cells that are cancerous from that tumour spread throughout the body, they form secondary tumours elsewhere. Okay, so this is our primary initial tumour of cancer cells. And this is where it is broken off. So this group of cells then would go on and form a secondary tumour. Okay, now people have probably heard about this before. If you have, say for example, a person with lung cancer, if that cancer and those cells detach from that original tumour, it can spread around the body and then lodge in the liver and attach in the liver and start to grow and continue to grow there. Okay, sometimes it's called metastasis. You won't need to know that term, but it's sometimes one that you might have heard of. All right, so that was a whistle-stop tour of your division and differentiation part of the higher human biology course. Okay, the next video will be on your DNA subtopic.